Prophase is going to be the first phase of meiosis following an interphase. And what is going on in this stage is the chromosomes are starting to form from where they were in interphase is kind of a coiled mess of DNA. They're becoming a more compact DNA and forming homologous chromosomes, which means they're going to be the same length, and the centromeres that are going to be these clear beads are going to hold them together, which they are going to be called centromeres. They are going to be formed together through a process called synopsis, which literally means bringing together, where you're going to have a paternal and a maternal chromosome from each are going to pair together and to form your larger chromosomes, and then your smaller ones. Also, during this phase, you're going to have your centrosomes, which are going to be your washnuts here. They're going to be starting to form, and there's going to be a spindle fiber also forming together too. Metaphase is the next stage of meiosis following prophase, where the homologous chromosomes are going to align in the equator of the cell. Also during this, the centromeres are going to migrate to each the north and south pole of the cell through the spindle fiber. One of the easiest ways I always remember metaphase when trying to study it is meta and middle is that the chromosomes are all kind of coming towards the center of the cell. Also during this, you're going to have a law of independent assortment where the chromosomes are going to kind of shuffle around, and in that shuffling, they're going to kind of move back and forth different genes of your DNA. So this is going to, be what would tell you if your child had blonde hair or is 6 foot tall, or just different genes that make each child unique. The next phase following metaphase is anaphase. In this phase, the homologs, which are each of the pairs of chromosomes, are still consist of two sister chromatids held together by a centromere. And these homologous pairs of chromosomes are beginning to migrate to opposite poles of the cell. Also, during this phase, the chromosomes are forcing the cell to lengthen and start the process of pulling apart. Telophase is the next phase after anaphase. And what's going on in this phase is that there's cytokinesis is beginning, which the cell is starting to form a second cell, but they are still conjoined as one cell. During this, each chromosome in each daughter cell, here and here, consists of two replicated sister chromatids. So they've separated, they've duplicated. The big ones have gone to one side, and the other one has gone to the other side. They're forming their own cell here. Prophase 2 is going to be the first stage of the beginning of meiosis 2. This is when your cells are going to split apart, and you're going to have two separate daughter cells. In each daughter cell, the chromosomes are going to begin to condense again in each daughter cell, just kind of like prophase 1. Here in metaphase 2, following prophase 2 and meiosis 2, the sister chromatids are all going to start to migrate towards the middle. Another easy way, remember meta, the chromosomes will all go to the middle, to the center of the cell, just like similar in metaphase 1. Also, the microtubules will attach to the centromere as well. During anaphase 2, following metaphase 2, the sister chromatids, rather than the homologous chromatids where they were all uniform and same size and same length, they have separated at the microtubes and pulled apart. So you're going to have each one, and when they get pulled apart, they are going to migrate to opposite poles of each cell. And last is telophase 2 and meiosis 2. After once completed, the sets of chromosomes are located at each pole. You're going to have cytokinesis occur, and after a cytokinesis occurs, both daughter cells in the past in anaphase 2, you're going to all separate, and you're going to have form 4 haploid daughter cells, and that is the final stage of meiosis 2.